After you calculate your electric fields in SimNIBS, you get the mesh files from SimNIBS. I'm gonna explain some of the things that you can do for post-processing in Matlab. Keep in mind that you can do all of these things in Python as well if you're more interested in Python. For this example, I have run like a quick random analysis of a TMS on a brain, and it really doesn't matter what exactly this simulation is. This could be TDCS as well, or TMS. It could be anything. But as you can see here, I have like the voltage, electric field, and norm E and the coil dipoles in the mesh file. And I want to explain how you can open this mesh file in Matlab first. So here's like a few lines of code that I have written. I should mention that SimNIBS comes with a set of Matlab functions and Python functions to interface Matlab and SimNIBS. And let me show you where those files are. So in Windows, like if you go to, or in any operating system, if you go to SimNIBS folder that you have installed SimNIBS on, you see a folder called Matlab that has a bunch of Matlab functions that you can use in your scripts. And I suggest you add this Matlab, like this folder to your path, so that you can easily use, like add that folder here, easily use those functions for your analysis. The first thing that you have to know is that you need to open the mesh files in Matlab. And this is the command that you use, mesh _ load _ gmesh4. And then you give the path to the mesh file that you want to open in Matlab, and that will open and put everything in a variable called mesh. It could be any variable. So if I run that line of code, you see it starts with running, like reading the mesh file, and it gives some brief like explanation of how the number of nodes, the number of triangles and stuff. And I'm going to explain that. And I want to also, there is another note that any function that you have, you can just open it. And there is a help file for that that explains what is the input files to each function and the outputs, and if there is any optional variables that you can specify. So first, let's go over the structure itself. So when you open any mesh file, you would see some variables, like, for example, triangles, triangle regions, tetrahedra, and other stuff. And as long as you understand how these structures work, you can basically write any script that you want that does whatever you want. And so let me explain what these variables are. Triangles is a matrix that explains, no, basically before that, I have to explain nodes. We have a variable called node, which is a matrix that is a 753,000 plus by 3 variable matrix. And if you open that node, you will see that each row corresponds to the coordinates, the Cartesian coordinates of a single node. This is X, this is Y, this is Z for node 1. And what I mean by node is, let me show you the mesh file in gmesh first. So If you zoom into the mesh files that you have, for example, I have opened the gray matter, you see it's made up of a bunch of nodes that are just dots connected with lines together, and 3 lines together will form a triangle. And this is like a mesh in the surface. And if I show you the volume of the mesh, you will see everything is connected together with 4 triangles and 4 nodes together and form a tetrahedra. And with that tetrahedron mesh, we can mesh the whole brain, like all tissues. And the way these nodes information are kept in Matlab is by having the Cartesian coordinates of each of these nodes in Matlab. So for example, node 1 corresponds with a point in space with this x, y, and z. Node 2 has another x, y, and z. After I have all the information in nodes, I can connect those nodes together to get triangles. And triangles refer to the triangles at the surface of each tissue. And what we see here is basically the index of the nodes. For example, triangle 1 is made from connecting node 3 to node 1 to node 2. And I know the coordinates of node 1, 2, and 3. If I connect these 3 nodes together, I can get my first triangle. And if I connect node 1, 4, and 2, I'll get my second triangle. That way I can have all the triangles on all surfaces this way by indexing the points that belongs to each triangle. And we see that we have this many nodes in our mesh file. Sorry, we have this many nodes and this many triangles in our mesh file. And each triangle belongs to a certain region. For example, we have like 6 layers in our model, like scalp, bone, CSF, gray matter, white matter, and stuff. And each of those like tissues have a boundary, a surface boundary that is denoted by these triangles. But in order to refer to each triangle to see how each of these triangles belong to what region, we can open triangle regions. So what we see here says, for example, triangle 1 belongs to region 1001. Triangle 2 belongs to 1001, and 1001 is the boundary for white matter. And if you go through like a different region, for example, you see this particular triangle number 669,622 belongs to the 1004 surface, which is the surface for bone, I believe. And this way you can index what each triangle is like made up of. And the same concept goes for tetrahedra as well. So triangles are made by connecting 3 nodes together. Tetrahedra are made by connecting 4 nodes together. And we see, for example, here, the first tetrahedron is made by connecting point like 515,151 to this point, to this point, to this point. And I already know the coordinates of each of these points in the nodes here. And by connecting each of those nodes together, I can form my first tetrahedron. And the same for all the other tetrahedrons. And so this way I can mesh the whole volume in my head model. And also for the same concept goes for tetrahedron regions as well. Each tetrahedron belongs to a certain tissue. And this is the number that corresponds to a certain tissue type. For example, tissue 1 is white matter, tissue 2 is volume for gray matter, and so on. And so far we've covered the the whole meshing inside the model, but now we have to go over how the information for electric field, voltage, and like current densities and things like that are kept into our like structure. So I'm going to start with the node data. So in SimNIBS, data is either kept at each node, like each of these particular points can have a variable, for example, voltage corresponding to it. And we also have element data. For example, electric fields are not kept at each point in our mesh, but they are kept at each element, which is like either triangles or tetrahedra. You can assume these values, the element data, for example, electric field, show the electric field at the middle of the triangles or at the middle of each tetrahedra. I'm going to start with node data, which basically has one structure in it because and if I open that, it says name V. So this means that this structure that I have opened corresponds to the voltage data in my head model. And I can see that I have variable data that has 750,000 and something variable shown in a vector. And if I look at my mesh in the beginning, I see that I already had 753,000 and something nodes. And like by these node data shows the value for voltage at each of those nodes. And then if I go back to like my mesh, I also have element data that includes variables like electric field and norm of the electric field or current density or any other element data that you have. Like for example, I have two structures because in my model, I have electric field and norm of the electric field. If I open each of them, it will show me the name of that variable. For example, this structure contains the values for electric field. And I talked about this like when we talk about element data is the variables at the middle of triangles or tetrahedras. And we keep each of those variables separately. For tri data is the variables for electric field at the middle of each of those triangles, and tet data is the variable that shows electric field at the middle of each of those tetrahedra. And if I open each of them, I see a matrix like n by 3 matrix. And this shows, for example, at tetrahedra 1, I have an electric field with X component, Y component, and Z component. And the same for tetrahedra, like in the middle of the first tetrahedron, I see an electric field is X component, Y component, and Z component, and etc. for all the other triangles and tetrahedra. And if I open the second, like let me go back, like if I have open element data, the first one corresponded to the electric field, the second one corresponds to the norm of the electric field, which is just the magnitude of the electric field. And again, I have tri data and tet data for triangles and tetrahedra. And each of them only have one variable now because magnitude is just a scalar. It's not a vector anymore. And if you actually calculate the norm of the electric fields at each of these elements, you will get the same results for the magnitude. So you can go from like electric field to norm E as well if you don't have it in your model already. Okay, now that we are familiar with the structure of the mesh files, we can do some post-processing analysis. For example, because I am interested in gray matter, let's say I want to extract the mesh files just in the gray matter. For example, the mesh file that I already opened has all the tissues. If I open them, for example, I can show a different tissue. So the mesh file that comes from SimNIBS has all the meshing for all the tissues. But because I'm interested in, let's say, just looking at gray matter, I can use this command to extract the information just for the gray matter. And this is the command line. Again, mesh extract regions. As the name suggests, it extracts a particular region that I'm interested in. And if you open like the help file or just the code, you'll see that it receives an input mesh and gives you an output mesh, and there are some optional variables here that I will explain in our demo. For example, because let's say I am only interested in extracting the tetrahedra from the gray matter, I specify like element type tet and region index 2. A reminder again, region 2 belongs to the volume of the gray matter. And I will extract the tetrahedra as the element type belonging to gray matter. And if I run this line of code, I will get another mesh file that, as you see, like is the same structure, but it's a bit smaller because it doesn't have as many nodes. It only has the nodes for gray matter. And for example, I can save that mesh file and take a look at it in Gmesh to see how it looks like. And the command that I use is mesh save gmesh4. And the first variable that I give is the mesh file that I have, the mesh variable that I just calculated, gm, and the name of the mesh that I want to save it as. If I run it, it will open, like it has written this mesh file for me. And when I open it, it takes some time to open. Okay, so as you can see, it has extracted the results for voltage, electric field, and norm E just for the gray matter. If I go to visibility here, I'll see that I only have volume 2. I don't have the rest of the tissues anymore. And let's continue with our command line and do some other stuff. For example, let's say I want to analyze the norm E of my simulation. What I can do is like first extract all the variables, all the values for the norm of the electric field in the gray matter and put it in a vector here and then do some stuff with it. And like this is another command that you can use, for example, that if I run this, it will tell me, like, let me show you. So as I showed you before, like if you open gray matter, like you have two types of element data. One stands for electric field, one stands for norm E. Let's say you want to write a like long script and you don't want to manually go over each of these element data and see whether the first structure belongs to electric field or the second structure belongs to the electric field, to the norm of electric field. What you can do is use this command to tell you which one belongs to the norm E. So if I run this, it will tell me that field index is 2, which means that the second structure in the element data has the norm E for me that I am interested in. And now that I know what index I have, I can just extract the information from there. I start with the gray, the mesh variable that I had for gray matter. I want to extract the element data. The field index corresponds to the norm E that I just found. And then I just want to look at the data at the tetrahedra. So I will say dot tet data. And if I run this, it will give me a variable that shows the electric field throughout the gray matter without any mesh information, because right now for this part of analysis, I don't care about mesh information. I just want to have the norm E at all the elements. And then I can do some like stuff with it. For example, I can calculate a histogram of the norm E variables that I found. And okay, so this is the default value, and I see that the bins are not small enough, so I specify like draw 50 bins for me, and it should show the, for example, the histogram of all the gray matter value, and we see like what we see is that as we expected, most of the values for norm of electric field is like belong to very low volts per meters, and only a few of them have high volts per meter value, like electric field values. And that is what we expect, because like if I look at the norm E, if I look at the norm E, I see most of the TMS is focused around this area, and most of the like major parts of the brain are blue. They don't have low like electric fields. For example, this is one way you can do stuff. Or let's say you want to look at the maximum electric field in your SimNIBS results. So this is a variable that includes all the norm electric fields in the gray matter. I just calculate max, and it gives me like, this is like 1.6 volt per meter is the maximum electric field that I have in my results. And keep in mind that I ran the simulation with the default DI/dt. You can scale it to the actual value that you're interested in. But another thing that I can do is, like, typically we would use like 99.9 percentile or something like that of the max to remove any like outliers in case you have in your results. And I can calculate, like, let's say here I have calculated a 99 percentile of the norm E in the gray matter. And if I run this, I will get this value for it. I can calculate mean or median, and it will show me all the results. But let's say I don't care about the electric fields throughout the whole brain, and I want to only study this red region that is, I call the affected area, like somewhere near the bottom of the coil, that we see most of the activity in that region. So how do I like extract that region? For example, because I define a threshold. Let's say I say extract all the values that has like half of the maximum of the electric field. Like here, if the electric field is 1.6, and I'm interested in all the areas that have like green, yellow, or red regions, which is like this region that I call affected region, I want to extract the values for that. I can easily just specify, get the index for the values that have higher than a certain threshold, and I like record that values and put it in gray matter norm E affected value, like affected volume. And I can calculate the mean, for example, of this affected volume here. Oh, sorry. And if I calculate, for example, we see that 0.6 volt per meter is the mean of the affected volume. And also I want to show you some other things, like a lot of this, there are a lot of built-in functionality put into these Matlab functions, so you can utilize them. For example, I can just use this command to automatically calculate some summaries for me. If you don't feel like writing scripts for yourself and you just want some quick results, if I run this command, it will run and it will give me some results. For example, it calculates the percentiles of the gray matter automatically for me. Like this is the 99.9 percentile in the gray matter, and also like vocality and things like that. Or if I use like this command, it will open a GUI and I can specify the mesh file that you want to analyze. And after running, it will show me some results as well to me. For example, it draws the gray matter for you, and like it draws that histogram that I showed you. And I understand that the thing that I showed you is redundant, but I wanted to explain how you can write your own script manually in case there is no built-in function like this. And there are a couple of more things that I want to talk about is, for example, let's say you want to look at like the electric field value at a certain location in the brain. You have like the coordinates of like your points that you want to look at, but you want to see what are the electric field values at those coordinates. Like this is another example. I can specify the coordinates that I'm interested in, like in a matrix, and each row belongs to a particular like point that I'm interested in calculating electric fields over, and each point has an X, Y, and Z. For example, I have picked a point like close to the center of the coil that has a high electric field, and I have picked another port exactly on the opposite side of the brain, on the other hemisphere. Like as you see, the X value is positive, not negative. And if you run this like this command, it interpolates the values at these points, and it will give you the like the electric fields at this location that you want to look at. Because this part takes some time to run, I will run it now and I will explain something else, and then after it's done, I will come back, talk about the results. And the final thing I want to talk about, I believe we had a question from yesterday. Someone, if I understood the question correctly, someone asked what is the difference in the electric field maps within a certain subject if you have two different montages. And I don't have any results to show you, but this is like a general few lines of code. You should double check it because I haven't checked it. But let's assume that you, for a particular subject, you run, let's say, TDCS for montage 1 and montage 2, and you have two mesh files like from SimNIBS. What you can do to find the differences in the electric field maps is just open them in Matlab, and then I am making like because the mesh files in both of these mesh like structures are basically the same. First, I copy my mesh file into the mesh, the new mesh that I want to analyze my data, and then just change the values corresponding to the electric fields and voltage and the things that I'm interested in. So because I want to find the differences between the electric field maps, the only thing that I have to do is to change the variables that belong to those like the element data that include the electric fields. For example, like I have to find the difference between each of those corresponding variables. Let's say like I want to subtract the electric fields over the triangle at like mesh 1 minus the same data over mesh 2, and that way you can find the electric field difference, and I write it in the same variable in my new mesh file. And just something keep in mind, if probably, depending on what you want to study, probably what you want to do is to first subtract the electric fields from each other and then calculate the norm of those calculated electric fields from the new electric field, instead of just directly subtracting the norm of electric fields from mesh 1 and mesh 2. Because like magnitude and substitution do not, you cannot change the order. First, you have to do the subtraction, and then you can calculate like a norm, like similar to here. I have the electric field, I can calculate the norm. And then you can save the data in a mesh file and just visualize that. Hopefully that answered your question. And the results that I showed you for interpolation has like finished, and I can, this is the last thing that I will talk about. And the end results is something like this. If I open, it will give me 3 cell arrays, and in each array, like it will tell me I have an electric field, and this is the electric field for point 1 and point 2. Because I had 2 points for like interpolation. And if I open another one of those, for example, this one, it will show me the norm of the electric fields at point 1 and point 2. And as you expected, point 1 is like close to the center of the TMS and has high electric field, and the other side of the brain doesn't receive much electric field. So I know this was just a quick overview of some of the things that you can do in Matlab or Python, but I hope I could convey like the whole point of the structures in Matlab, and you can play around with these variables and do whatever you want to do and do any kind of post-processing that you want to do. And like if there is any built-in functions, like from the functions provided with SimNIBS, you can use them, but you're not limited to them. You can write your own scripts, as I showed you here. For example, extract the tissue that you're interested in, do some analysis on top of it. And finally, I want to refer you to some of the examples in SimNIBS. You can use them to walk you through some of the scripts. Like if you go through the SimNIBS folder and open example files, it has some example files for analysis, like simulation analysis, optimization, and it will walk you through some of the codes. And that way you can learn more about the scripting for post-processing and things like that. Okay.
Thank you, Zina, for giving us a feedback. There is a quick comment from Axel Tilscher. When you want to know the difference of the field strength, then directly subtract the norm E values rather than using difference of E. So just directly subtract the norm values if you need to know.
Sure. Yeah, okay.
Yes, I think we're gonna share this example script with all participants. We will attach it to the email when we will send around the materials. Now the next demo will be from Katie Mantel, and she will talk about models of brains with pathology and an example of pediatric stroke. Katie, the floor is yours.
All right. So I'm gonna kind of be switching back and forth between my presentation and Matlab and some other things. So hi everyone, my name's Katie Mantel. I'm a PhD candidate in Alex Opit's lab. I do a fair amount of simulation work using TMS. So in this demonstration, I kind of want to explore what's possible with scripting, just kind of give you a flavor of what kind of things I do. And I want to introduce you to working in 3D space, working with the meshes and matrices. And so to do that, we're going to be building a simple TMS grid. And we can do a lot with grids. It allows us, in the clinic, grids are used to map TMS effects. And then in the modeling world, we can use it to explore how the electric field changes with the coil position. We've seen it used in previous talks during this session. It can be used for sensitivity analyses and mapping as well, and it also can be used in optimization. So quick reminder, we are working in 3D space, so we have multiple coordinate systems. We have the head coordinate system. We're building a grid that has its own X, Y, and Z. The coil has its own coordinate system. And then we're working with discrete triangle meshes, and each triangle, Sina did a nice overview of the nodes and how triangles are defined. And so we can also get things like center points for those triangles and the normal vectors of those as well. And of course, that does mean that a triangle has its own coordinate system. So quickly, our first goal of putting a TMS grid on is to align our grids to a center point. So we can choose any point on the head, and we want to place a grid centered around that point. So this might be the plane of the center point we've chosen, and this is the grid that we're trying to build. Basically, we get to use Rodriguez's rotation formula that allows us to rotate the grid Z vector so that it lines up with the center point normal. And ultimately, we're trying to get this outcome. So our grid is mapped onto our plane. So what that looks like in Matlab is this. So to begin with, we first load the mesh like Sina showed us previously, and we're going to extract regions. In this case, I'm interested in the skin surface because it's not invasive, so we're placing the coil on the skin surface. And we have a few in-house scripts, but they're pretty simple. So we can get the centers for each of the triangles. So if you take the coordinates of all of the triangle nodes, you can average them, and that will give you the center for each of the triangles. And then you can also find the normal vector for each of those. And then, so once we have our centers and our normals, we can then essentially figure out how what theta is needed, and then build a rotation matrix and apply this rotation matrix to the grid. Now, I didn't show at the beginning is that we do define an initial grid X and Z coordinate. This is for having the grid straight on. If you want it to be tilted any bit, you would put in a different set of values here. Okay, we'll go back to slideshow. All right, next we actually have to get the grid points, what the values are. So if we think about the grid, the center point that we chose, we can think of as being 0, 0, 0 on just a regular XY plane. And then we can choose our spacing that we want. And so then each of these other points can be defined relative to this point using our spacing. So a few examples, if we move in the negative X direction, our X value will be minus S. Or if we have another column here, it would be minus 2S. And if we move in the positive direction, now we have positive S for these columns, and over here would be 2s and so on. Same for y. But now if we rotate our grid so that we're looking at it straight down, it becomes a little more complicated because we're trying to map this to a curved triangulated surface. And so if each of these yellow line segments are a triangle and then the blue dots are the centers, we're trying to have it curve along the head. So actually what we're doing is we're mapping our estimated coordinates, which we got from our spacing, and we tell it to find what's the closest triangle center to this point, and that's going to become our actual coordinates that we use. One other piece to note is that we're using matrices in Matlab, so indexing is like this. So this top left is indexed as 1, 1, and so that's kind of the origin of a matrix, whereas over here we indexed at 0, 0. So it's just one thing to keep track of. Just make sure you know where you are in this grid and how you want it to show up in this matrix. And so that is this section here. So we have a couple for loops. And so I'm looping through. I've decided to keep it at my center point, the 0, 0 point. And so I'm looping from, in this case, -1 to +1 in the x direction and also in the y direction. And that means that my matrices in Matlab, I have to do some conversions into the index, the 1, 1 or the 1, 2 indexing there. Okay. The next thing that we need to do is we need to rotate the coil Y vector so it points in the direction that we want it to point in. And so there's many, many different ways of doing this. There were many ways of defining a grid as well. This just is one way of doing it. So I've decided to define everything based on the head Z vector. And so what I first do is I project the head Z onto the coil plane. And we do that using a vector rejection equation. And so that gives us this vector here. And then I again use the Rodriguez rotation formula and have it rotate by whatever theta I want it to. So typically when we're talking about particularly the motor area, we're talking about a 45-degree angle. So theta would be 45 degrees, and we're going to have it pointed in that direction. And then in this case, I'm using the expanded formula of Rodriguez's formula, just kind of to show that you can do the same thing in many different ways. So this time I'm defining my rotation matrix this way, and previously I had defined it this way. They're the same thing, but as with any type of coding, it all has to do with personal preference, and there's always multiple answers. The final piece is that I highly recommend checking out the SimNIBS website. Sina took us through how the meshes store information about triangles, tetrahedra, and then simulation results. And so that's going to be really, really important anytime we're trying to analyze data. And then Gmesh, which is the default visualization tool for SimNIBS, it allows us to create geo files to visualize our coordinates and vectors that we create. And there's some good documentation online for that. And so basically, once I've created my my grid on the coil plane and mapped onto the head curvature, I can then create a mat file that will run simulations. So I typically save a template. So I go to the SimNIBS GUI and set up an initial example of TMS. And then I save that as a template file, and then I can load it here and then change everything that I need to. So the folder locations for where my head mesh is stored, so I can figure out where the EEG cap is, what's the file name of the mesh, what fields do I want. So typically I'm dealing with electric fields and norm E, but Sina was also talking about V, and you can also store current and current density and things like that. One thing to note, open in Gmesh, when you run from the GUI, it automatically opens up the results. If you need to run this on a server or don't want it to show up after, if you're running like a bunch of simulations at a time, set this to 0. And then the key point is the position list. So what we're gonna do is essentially just loop through the number of positions that we have, which, and just add the coordinates. We have to add the vector, the X, Y, and Z vectors that we calculated above, and then place it at a position which we calculated based on the head curvature. From there, we can save the structure that we just created as a simulation file. It's just a .mat file. And so later on, we can just use SimNIBS from the command line and call this mat file. And I always save the information about my grid locations as well as my coil vectors. That way I can do analysis with that information later on. And then these, I think, are more in-house scripts that we have, but they're pretty straightforward. We're just giving it a list of points, so coordinates, and what value we want there, and what file name, and it's just going to make sure that it's in the correct format for Gmesh, and so it will allow us to do visualization. So I guess I've chosen a position that's for C3 on Ernie. And so I've given it all of the information about where to find my Ernie mesh. And so if I run this, it goes through all of that. And then this is Ernie. And so I can go to, I just created this folder, so this is from today. And here are all of the files that I just created from running that Matlab code. And I'm going to be interested in choosing the position numbers, and then we can also look at the vectors. And so you can see this was the center point that I chose for C3, and all of my points are lined up nicely along the curvature of the head. If we look at the vectors, this is the, I guess I can move that up. The Z vector. So this was what normal to this location, and so if we flip that, that's how it points into the head, and that'll be how we orient the coil plane. And then this is the Y vector, and so you can see they're all pointing approximately 45 degrees, and that's the goal. Later on, you can run that in SimNIBS. So I guess if we open, this is the file that I saved. So this is the simulation file. You can open it up right in the GUI, or you can do this from the command line. Sorry, my computer is a little slow right now. Okay. The position list is not showing up right now, but usually you'd see the position list and it will have the coordinates of all of the grid points. But I don't know, my computer's quite slow right now, so sorry about that. Yep, so that is kind of how we go about building a grid. And then there are a lot of ways to expand this. This was just a very simple small grid, 3 by 3. If you want to make the grid size bigger, there are things you'll have to take into account, like more head curvature. And if you span both hemispheres, you have to make sure that your negative signs are accounted for. The Coil Z Vector, I've kept it very simple as just the normal of the position of the coil, but that can lead to a lot of variation just because the skin surface isn't completely smooth. So you might want to look at a larger area and take more of an average. And then if you're interested in choosing the same center point across participants, there are things you can do like transforming coordinates from M and I space, or you can use the EEG positions that HeadReco, for instance, provides. And so there's a lot of different things to expand this grid. But that was just kind of a basic overview of how to build a simple grid. So yeah, I will pause there. Are there questions about that section? Nope. Okay.
Well, I'd like to just chip in and say thanks. It's Axel, because we get really a lot of questions about how to define TMS calls and orientations, not in the graphical user interface, and you implicitly covered that by explaining how to define now how to set up this rotation matrix. So thanks a lot for that. That will be super useful to have. In case, I also want to just briefly mention, in case you just want to have a TMS call optimized, then we heard today a very nice presentation from the Peterchev lab, but there's also our TMS optimization, which indeed does more or less the same under the hood. It spans up a grid and then does that for you. But indeed, often we have questions on how to define a coil orientation and how to what the heck this matrix means. That's super useful to have now. Thanks.
No problem. Yeah, it's definitely a lot to wrap your head around working in the 3D space. So yeah, I like having visualizations for that. All right, so the next piece that I'm going to talk about is non-standard modeling, and I'm going to be focused on pediatric stroke. So just a recap of how models are built for SimNIBS is we start with the MRI. The T1 is necessary, the T2 is optional, but highly recommended to get an accurate skull and CSF boundary. Then using something like HeadReco, you can get the masks for the different tissue types. So in this case, I'm just showing gray matter and white matter masks. And from there, SimNIBS takes care of all of this, but it makes the surfaces, so just the triangulated surface, and everything is nested within each other. So it's a little hard to see, but white matter is completely enclosed within gray matter, which is completely enclosed in CSF, and then bone and skin. And then the meshing process occurs where the tetrahedra are kind of backfilled in from between the two surfaces. And so everything from this outer gray matter surface to the inner, white matter surface is filled with gray matter, for instance. And that's why for this type of model building, it's really important to have all of your surfaces properly nested. Now, when we're talking about stroke, which, as most of you, I assume, know, it's a blockage of oxygen to the brain, and a lot of the cells can die off. And then, depending on where that lesion, where the cells died was, can have different impairments. And so, as the non-invasive brain stimulation community, we're really interested in using that to help with recovery and help these individuals have the best life as possible after such an unfortunate event. And we work with Dr. Gillick's lab, who does perinatal stroke, and so it's an estimated 1 in 3,500 to 1 in 2,700 live births are affected by perinatal stroke, and it can result in things like cerebral palsy. So the problem is, when we're looking at these cases, is that HeadReco and other auto segmentation routines were built to function with pretty standard adult head MRIs, and so it can lead to issues in the masks. And so here, this is the lesion, and it's saying that it's filled with gray matter, which is not the case. So we have to fix this by hand. Our group tends to use ITK Snap, which is an open source free software that you can download. And so it allows us to open up a T1. So we're going to use our T1 conform that HeadReco created. And then we can open up a segmentation mask. And so in this case, for instance, the gray matter. So the gray matter nifty file that HeadReco created. And now we can see that, we can scroll through all of the different slices. And we can see that clearly, this was done incorrectly. I also really like ITK Snap because it has a 3D visualization, and so you can see what the surface looks like, which is sometimes hard to imagine when you're just working in slices. For hand segmentation, it can be tedious and it can take a long time, but it's really important to have these accurate meshes when we're talking about figuring out where the electric fields are. And so we can play with a brush. And so if we kind of zoom in here, and so we see that this area is filled in where it shouldn't be. We can take our brush, we can basically choose that we want to clear the label, and we can remove this. And so you can play with brush size. You can also play with polygons. And so instead of brush, you can kind of be like, I don't want anything in this region, and it gets rid of everything inside of it. Sometimes, because these are children with stroke, sometimes it doesn't always get all of the gray matter either. So we can do kind of the opposite thing. So we can put it back to label 1 and come in and be like, okay, all of that is really gray matter, so I want that to be covered. And then you can see that for one slice, we were able to remove that there, and over here we can just keep removing the sections that we don't want. So as you can imagine, going through, we have hundreds of slices in all of the directions, so this can take a while. And so I have found that Hedrico can handle small lesions pretty well, but the larger lesions can be pretty tough and require a fair amount of hand segmentation to fix. But once you have that, you can then save this new mask that you've updated. I'm not going to do that right now, but you can save it. And then we go to SimNIBS website. If we go to Hedrico, yesterday Ola was telling us that Hedrico can be used with all, which does all of the steps, but there are ways for manual editing. And so we can do Hedrico surface mesh. So if we fixed some of our meshes, we can do surface mesh and volume mesh. However, sometimes if you're using Cat 12, you're going to have to figure out the steps yourself, which is a pretty involved process to make sure that all of the surfaces are properly nested and it will be able to backfill. But that is kind of the overview of how you would go about it. It does take time. It's pretty time intensive. I would highly recommend, if you're going to be doing a lot of these, to get a tablet and stylus. It makes it 100 times better, and then just listen to some music while you're segmenting. But oh, I did forget to show you. So this was the original surface of gray matter with this error in the segmentation, and this was my final version of it that did not have that sticking out into the lesion, and so it's now accurately modeled. And so then I can run simulations with that, which we can talk about real quick. So once we have our finished lesion model, we might want to ask some pretty obvious questions. What happens when we stimulate right over the lesion versus what happens when we stimulate over the intact cortical tissue? And if you think this is the perfect time to use the grid that we just built, you would be absolutely correct. And so we can choose a point here that's kind of the center of the lesion and then build a grid around it. If you'll notice, my grid is quite a bit larger than a 3x3. And so this grid was not built with the code that we just went through, but it was similar, and that's how I started. And when I found there were issues as I got into these back portions that are curved more away from here, I made some updates based on that. And so we get our final grid on the surface of the head, and then we can run simulations for all of these. And so this is just an example. As you move the coil, you can see the electric field changes, particularly over the lesion. You know, with there being no gray matter tissue there, it changes where the electric field is located. And so this is just a preview of some of the results that we're looking at. So we're writing a paper right now, but basically we're finding that the lesion is affecting the average electric field when you look at, you know, the sections over the lesion compared to the non-lesioned hemisphere. So you look at the equivalent points over here. And so this is the lesion side. We get a lot, our average kind of drops compared to these mirrored points, what you would expect it to be if there were no lesion there. And we also get more variability on this side. So we get some higher highs and some lower lows. So it's pretty interesting stuff. I think it's pretty cool. And with that, just our collaborators over in Dr. Gillick's lab have been great with the pediatric stroke, and then all of my lab mates are awesome. So thanks.
Thank you, Katie, for great introduction into how to do grid stimulation and how to work with lesion brains. There are more questions in the chat that are coming, and I encourage you then to answer them. And for the final parts, I will continue this demonstration with talking about the animal models and translational models. And first, I would like to start with why we want to do that and what we saw. So we all know what is transcranial electric stimulation, how it affects human brains. We had many great talks through the course of this workshop. However, in order to truly understand how the stimulation operates in the brain, we need to be able to record what's happening in the brain, which is invasive procedure. Therefore, it's hard to do in humans, and that's why animal models are so crucial and important for us, because we can model all sort of conditions there and get direct recordings. However, there is a problem that animal brains are substantially different from human brains in size and in many other aspects. Therefore, it's not really straightforward to translate stimulation conditions from animal, like laboratory animal, like mouse or capuchin or monkey, to human and other way around. Computational modeling can help you with that, because you can model conditions that you would like to explore in humans and then make a similar model in animal and see what parameters you need to tweak to maximize a comparability on the level of the electric fields in the brain. And recently we did such sort of research in mouse, monkey, and human. And you can see examples of the model on this slide right now. And we explored TMS and TDCS, and let's start with TMS. And the first question that we can ask themselves, what is the intensity of magnetic stimulation given the exactly same input, and what is the focality of stimulation. On the slide, you can see one of the stimulation conditions over the left central brain, so approximately motor cortex in humans, on monkey and in mouse, monkey, and human. And what we can see that, and what we can quantify is that for the same input and for the same coil, we see very different affected volume and very different peak magnitude. Paradoxically, with the higher stimulation intensities, in this case in monkey, than in mouse and humans. There is no secret why it's so. I will explain in a moment. But what we can do, we can also then select multiple coils and see what, if there is such a combination of stimulation intensity and coil size and coil orientation that will generate intracranial electric field that are comparable to themselves as much as possible, and therefore effects of the stimulation will be more comparable, and we can deduct certain information about the mechanisms of stimulation. We can also explore this on a surface in terms of components, so tangential components versus perpendicular components. We already heard why it's important, because these components can have different effects, as perpendicular component is the one that goes perpendicular to the surface and therefore parallel to the pyramidal neuron in the brain. And because of that, it has maximum affinity with these pyramidal neurons and has maximum driven power or maximum effect. We can explore in more details what will happen if we try different coil orientation or different stimulation locations. But for magnetic stimulation, the main dependency of maximum electric field and focality or volume of stimulation approximately the same. Obviously, for small mouse, stimulation volume of the brain is very high in comparison to monkey and human, and the maximum strength of the electric field induced by the stimulation by the standard coil is approximately same in mouse and in monkey and human, much lower in mouse, and there is a drop for human if you use smaller coils that are sometimes typical in animal research. The reason for this drop is that, and we can explore this analytically, is that first the intensity increase with the growing head size from mouse to monkey, because the bigger is the brain, the higher proportion of magnetic influx it can capture, and the stronger the magnetic field. But at some point, when the brain start to be much bigger than the magnetic coil, the density of electric field will go down, and therefore the highest stimulation intensity will also be lower. Similar research can be done in transcranial electric stimulation, where we also explore number of montages. This is one montage, left to right stimulation, temporal stimulation, left temple to right temple. Again, same electrode size, same stimulation intensity, but we can see that the mouse intensity is much higher inside the brain than in monkey and in human. Therefore, this sort of calculation will allow us to rescale. For example, if you want to research what will happen in the brain during the typical human TDCS session, which use about 1 milliamp or 2 milliamp, we can model this condition, we see the number. Let's say for 1 milliamp, it will be approximately up to 0.4 millivolt per millimeter. And we can say, okay, if you apply the same 1 milliamp in mouse, how much electric field will use, and this will be like 80 times more. So in order to generate the same electric field, we will need to apply intensity that is like 80 times lower in mouse than in human. And once again, we can explore all sorts of questions, including focality and amount of brain that is affected, as well as relationship between tangential and perpendicular components. Obviously, more gyrificated human brain have very different distribution of tangential perpendicular component than smooth mouse brain, and all these questions can be deducted. But for now, I want to demonstrate to you what exactly we can do and how you can use these results in your own research and how you can move forward from there. First of all, recently we shared the finite element method models that we prepared for the publication I just showed you. We will send a link, and it's already available online on Zenodo. You can go there and download the models for monkey and mouse, as well as a model of a slice, a brain slice, that is ready to be used using SimNIBS or any comparable software, because the models are in a standard format. And you can find all details here. For the visualization today and for the demo, I already downloaded them. Actually, I uploaded them there, but I also downloaded them. So it's working. Once you download them, you can start with your own modeling. But before that, I would like to say a few words about how exactly you can generate your models if you have your own data, and what would be the steps. As was already mentioned many times before through the course of this workshop, there are multiple principal stages that you'll need to come through in order to create a model. First, you have an anatomical image. Then from anatomical image, you go to the segmentation, so you define what part of the image does what. Then from segmentation, you go to the surface, and finally from the surface, you can go to the mesh. So on example of a mouse, once again, I will use ITK Snap. That is free tool that allow you to do segmentations. And this is a model of a mouse, DigiMouse, that actually was kindly provided and shared by the group of University of Southern California. They also did some anatomical labeling, which was very helpful for our case. But let's say you get your own image. That's what you will see. Unfortunately, for most animals out there, there is no automated pipeline how to do segmentation. So if in case of human, all these steps are pretty much standardized. In case of animal, like in case of mouse, you will need to segment everything manually, most probably, unless you use some very specific software and atlases. This point was already covered by previous talk of Katie Mantel, but I will just say that you need an image. It can be T1, T2, can be CT, can be a very accurate pencil drawing. It doesn't really matter at this point, as long as you can capture all elements of anatomy. And then you need to manually segment everything slice by slice, where each tissue that you would like to model will be labeled with a separate, identity. So you have label 1, label 2, and so on. Just more accurate than that. What's important if you plan to use a SimNIBS, that there are convention of labeling inside. This convention is explained together with shared models and also explained in SimNIBS tutorial. But I'll just say that these labels are crucial. So you always have label 1 for white matter, label 2 for gray matter, and so on. And labels start with 1000, 1001, 1002, and so on for the surfaces of for triangles. Once you have the segmentation, you can use FreeSurfer or any other tool to tessellate the surfaces into the tessellate the segmentation into the surfaces. It's literally the one command. You can put it here. It's MRI tessellate if you use FreeSurfer, and you supply your segmentation, and you'll get surface that you just need to convert to some standard format such as STL. Once you have that, you can also take a look on the surface, on all of them, to see how they look like and to see if they are all accurate. There are multiple tools that you can use for simple visualization of standard surfaces in a format like STL. Especially there are a lot of tools nowadays because it's exactly the same kind of task and same kind of problem which you will do in 3D modeling, or in 3D printing, sorry. And you can use pretty much any tool for that. I personally prefer lineup of Autodesk, and right now on the screen you see the simplest of Autodesk tools that I will show today, which is Mesh Mixer, that allow you to take a look on all the surfaces that you just generate after the segmentation, to see that they are accurately aligned, and to ensure that your surfaces, first of all, are enclosed, so there is no holes in the surface, so they're enclosed within itself, within each element. And second, they are not crossing each other, so there is no intersections. When you run SimNIBS, automatically there will be a way to manually, there will be a way to automatically correct for that. But in your own in your own animal work, you maybe will need to take a look on your surface and ensure that it's correct. And just for the demo, I can show what is the quick fix that you can do if there are some problem with the surface. I created a brain surface that is not really accurate so far. So there are a few things that you can try to check if there are some abnormal triangles there or abnormal elements. You may want to go back to your segmentation and double check that everything is accurate, or you may quickly work with the brain surface, as well as you can apply some simple. You may want to apply something simple like smoothing of your surface, or a meshing, or number of other operations, or analyze its physical size as well as alignment relative to the center of the region. When you work with a model, it will be important to make sure that all your surfaces are correctly aligned relative to the center of a region of the space. In this case, this is a red ball. So here you can see that, and maybe slightly correct that if need be. Once it's done, the next step will be meshing. Again, You can read more about meshing if you will go to the documentation of SimNIBS. And for example, routine head recall have its own instruction how to mesh from the surfaces, or you can explore what other options you have, and probably you will use standard functions such as gmesh. Again, this is just one line that will mesh your surfaces into the model using the settings that you will supply in a separate file. Once the mesh is done, so that is the files that we shared here. This is a brain mesh. This is a whole mesh of the monkey and the mouse, including brain. You can just download them and use. All other procedures are exactly as you would typically do. You open SimNIBS, you search for your model and add it here, supply some path for output folder, make sure that in stimulation options you don't try to interpolate it to NIBS or transform to an iSpace, because all these functions assume that you work with a human brain. And then double check conductivities. When I said that it's very important that when you do segmentation, you label everything correctly. This is the reason why it's important, because here working memory, white matter is actually any volume that is defined with the label 1, and gray matter is the volume that is defined with the label 2, and so on. So in order to make sure that conductivities are accurately supplied, you need to make sure that you also accurately label model. Once it's done, you set up a standard electrodes, standard position in a way how you want to do that. If you want to do that more accurately, you probably want to measure your real animal and estimate where physically you position electrodes relative to some anatomical points, and then you can go to a surface viewer and estimate the physical size of this model and see where the points will be. Yeah, so this is an accurate measurement of the model in millimeters. So you can find what will be the position on this model, again in millimeters, and position your electrodes same way. I already did that. I would supply that. Once you click run, it will run. You will receive the results in your output folder. And once you will open them, you will see what will happen. Here's our simulation. This is electric current on the surface of the skin. You can see it goes from electrode on head to the electrode on belly, and we can analyze what is the current intensity on the brain surface and how it's compared to the current intensity you would expect to see in human or in any other animal that you're working with. So this is a brief introduction into animal modeling. Once again, I encourage you to check what we shared and feel free to contact us in case of any questions. And of course, way more details about how it's done was written in the paper that is already published. Then I would like to conclude our demonstration and give word to Alex.
Right. Yeah, thanks so much for everyone holding out until the very end. So I hope we could give a good overview over some of the say more like advanced methods in SimNIBS. If you want to use some scripting, which is very helpful if you do more than one simulation, you want to write some small scripts. So I think we gave you some pointers to start that, or if what to do if then do non-standard models, how to put that together. I see Axel popping. Mark, do you want to also say something?
I think it was so far, I guess, at least from my perspective, one of the best workshops so far on SimNIBS, simply because it was very covering a lot of grounds. So I think it's a great starting point, and we should just try to continue making good resources out of that. Thanks a lot to you guys.
I feel like it went surprisingly well to do that over Zoom. From everyone's from home at different places. So I think you still got a lot of great talks and demos, and we are also planning, I guess you can already say that, to have that as a recurrent meeting. So next year, probably around the same time, we plan to do that again, and maybe let's see, hopefully in person. But we will see. I mean, the online worked also very well, so might be the future of scientific conferences. So yeah, that's for me. Thank everyone, and then yeah, hope we see everyone in some way soon again at some other conferences. And if you have any questions, need some help, feel free to reach out to us. Happy to help you with your own own modeling projects.